Hi, everyone. I'm Lisa Cunningham. I'm one of the principal investigators here at NIDCD. By way of training, I originally trained as a clinical audiologist, and then my PhD is in neuroscience. And my lab at NIDCD studies hearing loss caused by therapeutic drugs, which is one of the things that I'll talk to you about today. This is the EARssentials lecture on hair cell damage and protection. So by way of introduction, you may know that hearing loss is the most common sensory impairment in humans. It is often caused by death of the sensory hair cell, which is the receptor cell in the inner ear. And in non-mammalian vertebrates, hair cells are often regenerated and can result in restoration of hearing and balance function after hair cell death. However, in the mature mammalian cochlea, there is no hair cell regeneration. And in the mature mammalian vestibular system, there is a low level of regeneration. However, it is not enough to restore function. So that means that in the mature mammalian inner ear, the result of hair cell death is a permanent hearing loss or balance disorder. So today I want to talk to you about some of the stresses that can cause hair cells to die, and they include genetic mutations, which I'm not going to talk about today because EARssentials contains a specific lecture on the genetics of hearing loss. So today I will talk to you about hearing loss caused by ototoxic drugs, hearing loss caused by noise damage, and hearing loss caused by aging. And for each of these, I'll walk through how the stressor causes hair cell death. And then for a couple of them, there are some recent major advances in clinical care on these topics. And so I'm looking forward to sharing those with you as well. So first, let's begin with ototoxic drugs. These are therapeutic drugs that are used to treat life-threatening diseases, and they have as a side effect that they are toxic to the sensory hair cells in the inner ear. There are two major classes of these drugs. We call these drugs ototoxic, oto meaning toxic, oto meaning ear, and toxic meaning poisonous. And there are two major classes of these drugs. The first is the aminoglycoside antibiotics. This is a widely used group of antibiotics that are used to treat gram-negative bacterial infections. And I'll tell you a little bit more about these in the next few slides. And the other major class of ototoxic drugs is a group of anti-cancer drugs that have a platinum atom at the core of the drug. And the most ototoxic drug in this class is an anti-cancer drug called cisplatin. So both aminoglycosides and cisplatin are toxic to sensory hair cells in the inner ear, and we'll talk a bit more in the coming slides about why that is. And exposure to these drugs results in hair cell death and thus permanent hearing loss. So let's begin with the aminoglycoside antibiotics. This is a widely used class of drugs. I've listed some of the family members of the aminoglycosides here. They include gentamicin, which is widely used to treat sepsis in neonates in the neonatal intensive care unit. Neomycin, which is the active ingredient in the topical antibiotic Neosporin. So when you buy Neosporin and put it on your skin, the antibiotic in that drug is neomycin. Tobramycin, which is a drug that is routinely used to treat recurrent Pseudomonas infections in people with cystic fibrosis. Streptomycin is the first drug in this class to be discovered, and it is a drug that is used to treat tuberculosis. So these are widely used drugs that are used to treat a variety of life-threatening infections. These are among the most commonly used antibiotics in the world. And while you cannot buy them over the counter in the United States, you can buy them over the counter in most of the world. So in most of the world, people can treat themselves with aminoglycosides without a doctor's prescription. As I said, they're used to treat gram-negative infections, including Pseudomonas infections, which are common in people with cystic fibrosis. And so a high percentage of people with cystic fibrosis have aminoglycoside-induced hearing loss. Same for tuberculosis. So the side effects of aminoglycoside antibiotics include ototoxicity, so they're toxic to the inner ear, and nephrotoxicity, so they're also toxic to the kidney. When we look at aminoglycoside-induced hearing loss, I don't know if this is the first time in the course that you will have seen an audiogram. So on the right here, I'm showing you a typical audiogram This is a behavioral measure of hearing that is commonly used by audiologists, and it's a little bit different from a normal scientific graph in that the low numbers are on the top of the y-axis here, and the high numbers are on the bottom. On the y-axis is how loud the noise is in decibels. And on the x-axis is the frequency in hertz. And this is the normal frequency range of human hearing, at least for the frequencies that are most important for understanding speech. And the percept of frequency is pitch. So you can think of the x-axis as representing keys on a piano, or pitch. And an audiologist will always mark red circles for the right ear and blue X's for the left ear. And so normal hearing is at the top of an audiogram, as you can see here. And what this audiogram depicts is a severe high frequency hearing loss. And this is very with normal hearing in the lower frequencies. And this is very typical of what we might see of an aminoglycoside-induced hearing loss. Aminoglycosides cause a significant hearing loss or balance disturbance in up to 60% of patients taking aminoglycosides. That's according to a recent report from the World Health Organization. Approximately 41% of people with cystic fibrosis have an aminoglycoside-induced hearing loss because they're treated with repeated courses of aminoglycosides to address their Pseudomonas infections. Here, aminoglycoside-induced hearing loss is caused by death of sensory hair cells in the inner ear. This hearing loss is usually bilateral. As you can see from this audiogram, it's usually worse in the high frequencies than it is in the low frequencies, and it is permanent. The onset of hearing loss from aminoglycosides can be delayed for days or weeks after the onset of therapy, so it isn't always possible to notice that there's a hearing loss and then change the drug before the entire course of aminoglycosides is finished. So for each of the ototoxic drugs, I want to tell you a little bit about how these drugs do their day job, because these are very important drugs. So when we ask the question, how do aminoglycosides kill bacteria? We go all the way back to the central dogma of molecular biology, which is that DNA is transcribed into RNA in the nucleus, and then that RNA is translated into protein in the cytoplasm. And this step of translation requires a cellular organelle called a ribosome, which in bacteria has two subunits, a 50S subunit and a 30S subunit. Aminoglycoside antibiotics bind the 30S subunit of the bacterial ribosome, causing the bacteria to be no longer able to translate RNA into proteins, and thus killing the bacterium because it can't make any proteins. So you might ask, why do aminoglycosides not prevent protein translation in us? And the reason for that is that instead of having a 50S ribosome subunit and a 30S ribosomal subunit, our ribosomal subunits look different. We have a 60S subunit and a 40S subunit, and aminoglycosides don't bind our ribosome. So they prevent protein translation in bacteria, but not in humans. So aminoglycoside-induced damage in the inner ear is very specific to hair cells. Aminoglycoside, when you look at an animal or a human that's been treated with aminoglycosides, you'll see a death of sensory hair cells, and most of the other cell types in the inner ear are undamaged. So this death in the inner ear is very specific to the hair cells. When we look at aminoglycoside-induced hair cell death, we note that the basal hair cells are always more sensitive than the hair cells at the apex, and that the outer hair cells are always more sensitive than the inner hair cells. So this image shows a rat that was treated with the ototoxic aminoglycoside antibiotic amikacin. And here you can see at the apex of the rat cochlea, we still see a normal set, one row of inner hair cells here, and 3 rows of outer hair cells are all intact here in the apex of this cochlea. In the middle of the cochlea, we see that the inner hair cells are still intact. However, some of the outer hair cells are missing. And in the base of the cochlea, where the damage is worst, we see that the inner hair cells are still intact, but most or all of the outer hair cells are missing. And this is a very typical lesion for aminoglycosides, is that it's worse in the base than it is in the apex, and outer hair cells are more susceptible than inner hair cells. So when we ask how aminoglycosides kill hair cells, the answer is we're not entirely sure, but I'll tell you what we do know. So earlier in the course, we've talked about hair cell mechanotransduction, which occurs through a mechanoelectrical transduction channel on the stereocilia of the hair cells. And what we know is that aminoglycosides enter hair cells through this mechanotransduction channel itself. So the mechanoelectrical transduction channel is unique to hair cells, and we know that aminoglycosides can enter through this mechanoelectrical transduction channel pore. And so the hair cells have sort of a unique portal for aminoglycosides to enter the cell. Once inside the hair cell, aminoglycosides appear not to be readily cleared from the cell, and this reduced clearance may result in higher concentrations of aminoglycosides in hair cells than occur in other cell types. We also know that aminoglycosides can result in formation of reactive oxygen species in hair cells, and in animal models and tissue culture models, a variety of antioxidants have been shown to reduce aminoglycoside-induced hair cell death. But once inside the hair cell, we know that aminoglycosides eventually result in apoptotic death of the hair cell and thus a permanent hearing loss or balance disorder. So we've talked a bit about aminoglycoside antibiotics, and I'd like to switch now to talking about the platinum-based anti-cancer drugs, including this drug, cisplatin. Cisplatin is among the most widely used and effective anti-cancer drugs in the world. It's used to treat a wide variety of solid tumors in both pediatric and adult cancer patients. I've listed some of the tumors here that cisplatin is used to treat. And cisplatin is the most ototoxic drug in clinical use. It causes a significant permanent hearing loss in about half the people who are treated with the drug. And for some specific cancers, the incidence of hearing loss is much higher than that. For example, it's very high in children treated for medulloblastoma, where approximately 88% of those children will have a significant permanent hearing loss. Also, children treated with osteosarcoma and neuroblastoma have very high incidences of cisplatin-induced hearing loss. So you can imagine how devastating this is for the family to hear first that their child has cancer, which is a devastating thing for a family to hear. And fortunately, the survival rates of childhood cancer are quite high. So that's good news because this drug can save a lot of lives. However, it means that the child will have a very high chance of having a lifelong hearing loss. And so there's an enormous need clinically for therapies that can protect the hearing of patients who are treated with this life-saving drug. Cisplatin-induced hearing loss, similar to aminoglycoside-induced hearing loss, is sensorineural, meaning it happens in the cochlea. It's bilateral, it's progressive, and it's irreversible. So how does cisplatin kill cancer cells? So I indicated before that at the core of cisplatin, there is a platinum atom. And this is the entire cisplatin molecule here. And what happens when cisplatin enters a cell is that it permanently binds DNA. So it will bind DNA on guanine nucleotides and form crosslinks of the DNA. This platinum DNA cross-linking is called an adduct, and the cellular DNA repair machinery recognizes a cisplatin DNA adduct as irreversible DNA damage. It recognizes it as a kink in the DNA helix that is irreversible. And that triggers the cell to undergo an apoptotic cell death. So cisplatin kills cancer cells by permanently binding DNA and triggering an apoptotic machinery in the cell. So how does cisplatin cause hearing loss? So we may have talked earlier in the course about the fact that the inner ear has a blood barrier that keeps most drugs from entering the inner ear. It's somewhat similar to the blood-brain barrier in that it keeps a lot of drugs that don't enter the brain from entering the inner ear. Cisplatin seems to be the exception to this because cisplatin does not readily cross the blood-brain barrier, but it does readily cross the blood labyrinth barrier to enter the inner ear. Once inside the inner ear, cisplatin is retained indefinitely. So most tissues and organs will clear cisplatin in a matter of hours. And a few tissues like the long bones may take several weeks to clear cisplatin. However, it's still cleared pretty readily. In contrast to all this, when we look at an animal or a human that has been treated with cisplatin, we can still see cisplatin present in the inner ear months or even years after the last treatment with cisplatin. So it appears that cisplatin readily enters the inner ear. However, it is not readily cleared from the inner ear, and it remains in the inner ear indefinitely. This retention of cisplatin may result in ongoing damage and progressive hearing loss, and it's not unusual to see a progressive hearing loss in a person who was treated with cisplatin. Unlike aminoglycosides, cisplatin is toxic to many cell types in the inner ear, and I'll talk a bit more about that in the next slide. Cisplatin results in apoptotic death of hair cells and other cell types in the cochlea. And in addition, cisplatin also kills supporting cells and macrophages in the cochlea, and both supporting cells and macrophages have been shown to be able to promote hair cell survival and also clearance of dead cells. So the fact that cisplatin kills supporting cells and macrophages may mean that may contribute to its increased ototoxicity, because these cells contribute to the survival and well-being of other cell types in the inner ear. So when we look at cisplatin-induced hair cell death, it doesn't look that different from aminoglycoside-induced hair cell death. Similar to aminoglycosides, the basal hair cells are more sensitive than apical hair cells, and the outer hair cells are more sensitive than inner hair cells. Also similar to aminoglycosides, if you continue the drug or you increase the amount of the drug, the damage can spread from the base toward the apex. So in these images, you're seeing the cochleas of mice that were treated with cisplatin, and the hair cells are labeled blue with an antibody against myosin 7A, and the green is phalloidin to allow you to see some of the structure of the tissue. And so here in the apex, you can see that the inner hair cells are all intact, and the outer hair cells, 3 rows of outer hair cells, are also intact. And in the middle of the cochlea, you can see that again, the inner hair cells are all intact, but some of the outer hair cells are missing. And in the base of a cisplatin-treated cochlea, you'll see that the inner hair cells are all intact or mostly intact, and all of the outer hair cells are missing. So as I indicated, unlike aminoglycosides, cisplatin is toxic to several cell types in the inner ear. It is toxic to hair cells. As I indicated, it's also toxic to macrophages and supporting cells. It also kills cells of the stria vascularis and cells of the spiral ganglion neurons. So while aminoglycoside-induced damage is specific for hair cells, cisplatin-induced damage results in death of a wide variety of cell types in the inner ear. I wanted to tell you about a recent major advance in the treatment of cisplatin-induced hearing loss. And that is that 2 recent multi-center studies have indicated that a drug called sodium thiosulfate reduces cisplatin-induced hearing loss in children. Sodium thiosulfate is FDA approved to treat cyanide poisoning. And it has long been hypothesized that sodium thiosulfate would reduce cisplatin-induced hearing loss, because we know that sodium thiosulfate has a thiol group that will bind cisplatin and will permanently inactivate it. So the first study that I want to tell you about is a study that was published in the Lancet in June of 2017. This study was of 104 children at 38 participating hospitals in the children's oncology group. So fortunately, childhood cancer is quite rare. And so large studies of childhood cancer usually are carried out by consortia of hospitals. And one of the major consortiums of hospitals in the United States that studies childhood cancer is called the Children's Oncology Group. So this was a study of 104 children at 38 hospitals as part of a Children's Oncology Group study in the United States and Canada. These 104 children, they took all cancers. They didn't specify a single cancer. And they gave the children their normal cisplatin therapy. And then 6 hours after each dose of cisplatin, they gave them sodium thiosulfate or placebo. And what they found is that they saw hearing loss. 56% of the children in the observation group had a significant permanent hearing loss. So over half the children who did not receive STS had a significant permanent hearing loss. But only 28% of the children who did receive STS had a hearing loss. So STS reduced the incidence of a cisplatin-induced hearing loss by about half in this study. You can imagine that it's very important when we think about a therapy to reduce cisplatin-induced hearing loss that the therapy not reduce the therapeutic efficacy of cisplatin. It's critically important that the therapy not keep cisplatin from saving the lives of these kids. So this study showed that there were no differences in event-free survival or overall survival between the kids who received sodium thiosulfate and the kids who did not. However, there was an unplanned post hoc analysis that was conducted after this study, and that analysis suggested that the sodium thiosulfate may have reduced event-free survival and overall survival in patients with disseminated disease. So that means that they had metastatic cancer, but not in the patients with localized disease. So this unplanned post hoc analysis raised some concern that sodium thiosulfate may reduce the therapeutic efficacy of cisplatin in children who had metastatic cancer. The second study was published in June of 2018, and it was very similar to the first study, except that it was conducted by the European equivalent of the Children's Oncology Group. So this was 109 children from 52 participating hospitals in Europe. They, instead of taking all cancers like they did in the American study, they studied one cancer. They studied children who had hepatoblastoma, and it was not disseminated. So this was localized hepatoblastoma that had not metastasized. They gave the same drug protocol, which is that they gave the cisplatin as they normally would, and then 6 hours after the cisplatin administration, they gave the sodium thiosulfate. And they saw a significant permanent hearing loss in 63% of the kids who did not receive sodium thiosulfate, but only in 33% of the kids who did receive sodium thiosulfate. So similar to the first study, the STS reduced the cisplatin-induced hearing loss by about half in these kids. This study showed no differences in event-free survival or overall survival between the two groups. So the conclusion from these two studies is that sodium thiosulfate halves the risk of cisplatin-induced hearing loss in children. So this is spectacular news because it means that for the first time, children who are undergoing life-saving anti-cancer therapy with cisplatin will have a therapy available to them that can reduce their chance of having a significant permanent hearing loss from their cancer therapy. So in response to these two studies, the FDA approved sodium thiosulfate for use in children with localized cancer, not with metastatic cancer, in September of 2022. So the remaining issues for sodium thiosulfate use include there still is no therapy for children with metastatic cancer to reduce their risk of hearing loss. We still don't know if sodium thiosulfate is appropriate for use in adults. Sodium thiosulfate still only reduces the risk of cisplatin-induced hearing loss by about half. So we would rather have a therapy that does a better job than reducing it by half, although this is still a very important breakthrough, and I don't want to undersell it, but we would like to see a therapy that eliminates the cisplatin-induced hearing loss. We also don't know if sodium thiosulfate is appropriate for patients receiving cisplatin therapy for other cancer types. So while there are a lot of questions remaining, I can show you that there are at least 7 currently ongoing clinical trials using STS for cisplatin-induced ototoxicity. Some of these trials are only enrolling children. Some of them are only enrolling adults. Some of them are looking at localized sodium thiosulfate. So that is to deliver the sodium thiosulfate directly to the ear instead of giving it systemically. The idea here is to reduce the chances that it would interfere with the therapeutic efficacy of cisplatin. And this is these studies are looking at various tumor types, various ages, and so in 4 or 5 years, we should know a lot more about whether sodium thiosulfate is appropriate for children with metastatic disease, whether it is appropriate for adults, and whether localized delivery of sodium thiosulfate is as effective as systemic delivery of STS, or maybe more effective than systemic delivery. So to summarize the section of this lecture on ototoxic drugs, there are two major classes of ototoxic drugs: the aminoglycoside antibiotics, which are used to treat gram-negative bacterial infections, and cisplatin, and a group of a few related platinum-containing anti-cancer drugs that of which cisplatin is the most ototoxic. Both aminoglycoside antibiotics and cisplatin are toxic to sensory hair cells in the inner ear. Cisplatin is also toxic to other cell types in the inner ear. Cisplatin, as I indicated, is retained in the inner ear indefinitely. And the recent update that I told you about is that sodium thiosulfate reduces the risk of cisplatin-induced hearing loss in children with localized disease. So we've talked about ototoxic drugs. Let's talk about noise damage. So when we think about the risk of hearing loss caused by exposure to noise, we know that the risk is related to how loud the noise is and how long a person is exposed to the noise. So you may have seen this kind of heat map before, showing you how loud different sounds are. For example, leaves rustling is a very soft sound that's about 10 decibels. A noisy restaurant is about 80 decibels. An ambulance siren is maybe 120 decibels. A shotgun blast is maybe 160 decibels. And we know that the U.S. Department of Labor and the National Institute for Occupational Health and Safety set noise standards for how loud and how long a person can be exposed to different noises as part of their job. And both of these organizations have labeled 85 decibels as the limit of safe occupational noise exposure. So that is about the amount of occupational noise exposure that you would have in a noisy restaurant. And so the federal government requires that if your job exposes you to noises above 85 decibels, that you have to have hearing protection and a hearing protection program in place for individuals in those occupations. So I added this little chart here because it was some things that are more familiar, and those of us who don't have noisy jobs may enjoy music, for example. And so I'm just listing here the equivalent noise levels for some other things that you may have been exposed to, like the symphony or a rock concert, which can be around 120 decibels, comparable to an ambulance siren. And so when we think about, I did look up the modern generation of AirPods, and those will put out 110 decibels. And so according to the federal government, the maximum amount of time that it is safe to listen to 110 decibels is less than 90 seconds. So don't be listening to your AirPods as loud as they will go. I urge you to protect your hearing when you are participating in any of these sports, or if you have a noisy job, or listening to music as well. So when an audiologist thinks about noise-induced hearing loss, we talk about a temporary threshold shift and a permanent threshold shift. So a temporary threshold shift is something that you may have experienced before. If you've gone to a concert, or you've been out shooting skeet, or you've run your lawnmower, or you've ridden a motorcycle, and you may come back and notice that after that period of time doing that activity, your ears may feel muffled. You may have some ringing in your ears may feel full, and your hearing may be decreased a bit. And but in the next few days or a few weeks, you notice that your hearing recovers and that muffled feeling goes away. So we refer to this as a temporary threshold shift. And we call it a threshold shift because when you go back to that audiogram that I showed you earlier, and you raise your hand when you hear the beep, the softest sound that you hear, that you respond to, we call that your threshold. And so what we mean by a threshold shift is that the softest sound you can hear has changed. And so when we look at this graph, this is threshold shift on the y-axis. So this is a noise-induced hearing loss, and the hearing loss gets worse as you go up on this graph. And what I'm showing you on this graph is an experiment that Kate Fernandez, who is an audiology clinician scientist at NIDCD, Kate did this experiment where she exposed mice to an octave band of noise at 100 decibels for 2 hours. And she tested their hearing before and after the noise. And so threshold shift is the difference in the baseline hearing test and the follow-up hearing test after the noise. And what you can see is that a day after this noise exposure, these mice had a significant hearing loss that was worse in the high frequencies than it was in the low frequencies. However, 2 weeks later, the hearing loss had all gone away and the hearing thresholds had returned to normal, had returned to baseline. So because the hearing thresholds recovered from this temporary threshold shift, they fully recovered. That is why we call this a temporary threshold shift. On the other hand, a permanent threshold shift occurs when the hearing thresholds do not recover. So in this experiment, Kate exposed the mice to exactly the same noise, but instead of exposing them for 2 hours, she exposed them for 4 hours. So the same loudness, but a longer exposure. And again, this is the hearing loss that you see a day after this noise exposure. However, unlike the previous exposure, 2 weeks later, after this noise exposure, the hearing had not recovered fully, and this hearing will not recover, and so this is a permanent threshold shift. So what do we see in the inner ear of an animal or a human who's had a temporary threshold shift? So for one thing, we may see breakage of the tip links that connect one stereocilium to the next stereocilium. These tip links can break in response to the noise, and over time they can be regrown, and so the temporary threshold shift is recovered in this way. Another thing that we can see in ears that have experienced a temporary threshold shift is a swelling of the stria vascularis, or the blood supply to the inner ear. When we see a permanent threshold shift, we'll see a different set of manifestations in the inner ear. For example, we can see a complete loss of hair cells, which would of course mean a permanent hearing loss. In the event of a blast exposure or a noise that's loud enough, you can actually rupture the sensory epithelium, which will result in mixing of endolymph and perilymph and result in a permanent hearing loss. We may initially see swelling of the stria vascularis the way that we saw in the event of a temporary threshold shift. However, after a permanent threshold shift, we will often see actually atrophy of the stria vascularis. And finally, in the event of a permanent threshold shift, we will see a loss or death of spiral ganglion neurons. So this is an animal that was exposed to noise, and the reason that this oval looks so dark is that the spiral ganglion neurons are stained here, and they're packed so tightly that you can't differentiate individual neurons. However, after noise exposure, you can make out individual neurons because many of the neurons in the spiral ganglion have been killed by the noise exposure. So when I was training as an audiologist, we thought that a temporary threshold shift was safe because we could see that the thresholds would recover to normal. However, in 2009, Sharon Kujawa and Charlie Lieberman showed for the first time that the noise exposures that result in a temporary threshold shift may actually not be safe at all. And so the experiment that they did was in mice, and it was designed to give the mice a maximal temporary threshold shift. That is, how much noise can we give the mice and still get the thresholds to recover fully. So the noise that they used was an octave band from 8 to 16 kilohertz, which is about in the middle of the range of normal mouse hearing. And you can see that after this noise exposure, the mice had a significant threshold shift at 1 day that recovered somewhat by 3 days, and by 2 weeks had recovered fully to baseline. So this is a temporary threshold shift. And what they noticed is that although the thresholds recovered, the noise resulted in a permanent loss of ribbon synapses in the inner ear. So earlier in the course, we talked about these specialized structures in hair cells called ribbon synapses that are that allow for a continuous release of neurotransmitter from hair cells. And these ribbon synapses are absolutely required for hearing and balance function. And what Sharon Kujawa and Charlie Lieberman noticed is that in response to the noise that resulted in a temporary threshold shift, some of these ribbon synapses were lost and they did not recover. So in this graph, they're counting the number of these synaptic ribbons per inner hair cell. And you can see in the green line the number that they see in the control mice that have not been exposed to noise. And in the mice that were exposed to noise, you can see that even though at 8 weeks the thresholds had fully recovered, the number of ribbon synapses never returned to normal, indicating that even though the thresholds recovered, the loss of ribbon synapses was permanent. And a great deal more research is going on about this, where audiologists, as soon as we saw these data, were thinking about the fact that clinically it isn't unusual to have someone come in and say, it's not that I can't hear, it's that I can't understand. I can hear your voice, I can tell that you're talking, but if I'm in background noise, if there's a noisy restaurant, if I'm riding in a car, it's hard for me to understand what is being said. And so there's a lot of research going on to try to understand the relationship between this, what is called synaptopathic hearing loss, or hearing loss that doesn't result in hair cell death, but does result in loss of these synapses, and clinical manifestations of hearing loss, like an inability to hear speech and background noise. So the summary of noise-induced hearing loss is that the amount of damage is related to both how loud the noise is and how long a person is exposed to the noise. Noise exposure can result in either a temporary threshold shift, where the hearing eventually recovers, or a permanent threshold shift where the hearing thresholds do not recover. And even a temporary threshold shift inducing noise can cause a permanent loss of ribbon synapses, at least in animal models. So we've talked about ototoxic drugs and noise. Let's talk a bit about age-related hearing loss. So hearing loss is common among older individuals. If you know someone who's 85, you probably know someone who has an age-related hearing loss. 2/3 of individuals over 70 have a significant hearing loss. And I'm showing you here some data from Judy Dubnow's lab. Judy has an ongoing longitudinal study of age-related hearing loss that's been going on for over 30 years at the Medical University of South Carolina. And so they have a lot of data on what hearing looks like at various ages. And you can see that the hearing loss, the age-related hearing loss, is usually worse in the high frequencies than it is in the low frequencies, and that it continues to worsen as individuals continue to age. So age-related hearing loss is also called presbycusis. And it's a complex hearing loss because it's not pure aging. And the reason that I say that it's not pure aging is that humans don't spend their lives in quiet. Every human who is old enough to have an age-related hearing loss probably has some history of noise exposure. And we know that humans are pretty bad at looking back and recognizing all of their history of noise exposure. Also, not everybody knows every antibiotic that they've ever taken, for example. So age-related hearing loss is often a complex mix of aging and a lifetime of noise exposure, and possibly other things like exposure to ototoxic drugs. We also know that age-related hearing loss is not just about hair cells. We do see a loss of hair cells in animal models and humans who have an age-related hearing loss. However, we also see changes in atrophy in the stria vascularis, which of course is responsible for producing the endocochlear potential, which is required for normal hearing. We also see a loss of spiral ganglion neurons, and we see mixed age-related hearing loss or mixed presbycusis, where you may see several of these things in the same aged inner ear. So the current treatment for age-related hearing loss is hearing aids, and they can be very effective. Hearing aids have come a long way. They can do a good job of amplifying the sound that someone wants to hear while suppressing background noise. They're not perfect, but they can do a very good job and be very effective. Hearing aids are also very expensive. They can cost up to $3,500 each. Most people need 2 of them. They're not covered by most insurance, and they're not covered by Medicare. They also don't last forever. Most people need to replace their hearing aids every 5 or 6 years. So this is one of the barriers. The cost is one of the barriers for people with age-related hearing loss to using hearing aids. We know that of all the people who could benefit from a hearing aid, very few of them have actually tried one. So these data show you that about 1 in 6 US adults over age 18 reports that they do have some trouble hearing. So they're aware that they have a hearing loss. About 28.8 million US adults could benefit from using hearing aids. However, only 1 in 4 US adults who could benefit from using hearing aids has ever tried them. And so several years ago, several different groups were looking at what are the barriers? Why are people who would benefit from hearing aid use not trying hearing aids? And 2 different groups, the National Academies of Science, Engineering, and Medicine, and the President's Council of Advisors on Science and Technology, were both taking a look at this question of what are the barriers to people being able to try hearing aid. And one of the barriers that was identified is that hearing aids could only be obtained by prescription. They're not available over the counter. And again, they're very expensive, as I indicated in the previous slide. So both of these groups recommended that the FDA look at setting a set of rules for over-the-counter hearing aids. You know that you can walk into your local drugstore and try on some reading glasses and see if the reading glasses are helpful to you. And this ruling means that as of October of 22, you can also walk into your drugstore and try a hearing aid that is designed for individuals who have a perceived mild or moderate hearing loss. And so the idea behind making over-the-counter hearing aids available is that it would lower the barriers for people to try hearing aids by allowing them to try a hearing aid that is already engineered for someone with a mild or moderate hearing loss. The other thing that allowing over-the-counter hearing aids creates is an opportunity for companies that are well known in audio products. For example, here's an over-the-counter hearing aid being offered by Sony, and here's an over-the-counter hearing aid being offered by Bose. And so these are well-known companies that are already making excellent audio engineering products that we're all enjoying and we're all using. And so the idea is that now that these over-the-counter hearing aids are available, a person who feels like they have a mild or moderate hearing loss can go into the drugstore and try these hearing aids for less cost and a reduced healthcare barrier. So we'll see in the coming years if this improves hearing aid use among people who could benefit from hearing aids. Another reason that it's important to increase hearing aid use is that several studies have shown that hearing loss is associated with an increased risk for dementia. So in the study I'm showing you here, this was a study from the Lancet Commission where they looked at risk factors that contribute to risk for dementia. And they looked for modifiable risk factors. For example, they're showing you here that smoking increases the risk of dementia, that social isolation increases the risk of dementia. And the modifiable risk factor that contributed the most to susceptibility to dementia was hearing loss in middle age. And so we aren't exactly sure why hearing loss is associated with risk for dementia, and I'll talk a bit more about that in the next slide. But the important thing from this study was that hearing loss in midlife was strongly associated with an increased risk of dementia later in life. So as I indicated, we aren't sure how hearing loss is related to dementia. And this is an area of very active scientific study. But there are several hypotheses that are currently being advanced. The first is that hearing loss can make the brain work harder, forcing it to strain to hear and fill in the gaps. So people with hearing loss will report to you that it is exhausting to go through their day and to have to strain to hear what people are saying and to make out what people are saying. And the idea here is that the dementia community for a long time has been talking about cognitive load. That is, how much of your brain are you using at any one moment to achieve what you need to achieve? And so the idea here is that the hearing loss can use up all of the cognitive reserve and not leave much cognitive reserve left over for other tasks, contributing and thus contributing to dementia. Another hypothesis is that hearing loss causes the aging brain to shrink more quickly. And a third hypothesis is that hearing loss leads people to be less socially engaged. And we know that hearing loss contributes to social isolation. And so and we know that social isolation and loneliness are risk factors for dementia. So the idea behind this hypothesis is that hearing loss leads people to be less socially engaged, and so the brain is less engaged and less active, and thus increase the risk for dementia. So again, we aren't exactly sure what are the relationships between hearing loss and dementia, but several studies have shown very strong associations between hearing loss and dementia. And so I'm showing you a recent study here published in January 10th of 23, where hearing aid use is associated with reduced risk for dementia. And in this study, they used a nationally representative data set from a large ongoing epidemiologic study called the National Health and Aging Trends Study. This particular study included over 2,400 individuals aged 65 and older. The prevalence of dementia among the participants with moderate to severe hearing loss was 61% higher than among participants who had normal hearing. So this showed a very strong association between hearing loss and dementia in these older individuals. And among those 853 participants who had a hearing loss, if they were using a hearing aid, they had a 32% lower prevalence of dementia. So this study shows, one, a strong association between hearing loss and dementia, and two, an association between hearing aid use and reduced risk of dementia. There's an ongoing study right now at Hopkins being directed by Dr. Frank Lin, called the Aging and Cognitive Health Evaluation in Elders, or ACHIEVE study. And this is a randomized placebo-controlled study that is meant to be definitive to help us definitively determine whether hearing aids can reduce the risk of dementia. So this study is currently recruiting. It's going to recruit 850 cognitively normal older adults ages 70 to 84 with hearing loss. And these individuals are being randomized into a hearing intervention group, and this group will receive a hearing needs assessment, fitting of hearing aids, and education and counseling and how to use their hearing aids. The control group will not receive hearing aids, but is receiving an intervention called a successful aging intervention. And in this intervention, these individuals will have individual counseling sessions with health educators covering how to age healthily, how to eat healthily, how to get some exercise, and in general, how to improve their chances of healthy aging. So these two groups are being followed for 3 years, and the outcomes of the study include assessments of cognition, social functioning, physical functioning, and quality of life. So this is a very important study that is really designed to definitively determine the extent to which hearing aids, hearing intervention can reduce the risk of dementia. And results from this important 3-year randomized study are expected in 2023 or 2024. So I'm very excited to see what the outcome of this study is. So to summarize the section of the talk on age-related hearing loss is both common and complex. It's not pure aging, and it also involves whatever the person's history of noise exposure is, their history of treatment with ototoxic drugs, and possibly some genetic components as well. We know that hearing loss is associated with an increased risk for dementia. We know that hearing aid use is associated with a decreased risk for dementia, and I just told you about a randomized control trial to determine the extent to which hearing aid use can reduce dementia. And I told you that some types of hearing aids are now available over the counter. So we will soon begin to see studies looking at whether over-the-counter hearing aids are improving access to hearing health care, and whether people who otherwise would not have tried a hearing aid will now try over-the-counter hearing aids. So we've talked today about 3 different causes of hearing loss. We talked about ototoxic drugs from the aminoglycoside antibiotics and cisplatin. We talked about hearing loss caused by noise damage and how that can result in a temporary threshold shift or a permanent threshold shift. We talked about the fact that the amount of noise damage is related both to how loud the noise is and how long the person is exposed to the noise. And we talked about the fact that even a temporary threshold shift can result in a permanent loss of ribbon synapses in the inner ear. And finally, regarding aging, we talked about the fact that hearing aids are now available over the counter. That we talked that hearing loss is associated with an increased risk for dementia, and that treatment of hearing loss is associated with a decreased risk of dementia. And we should have more definitive results on the relationship between hearing aids and dementia risk in the next couple of years. So I thank you for joining me for this EARssentials lecture. I look forward to talking to you in the small group, the shorter lecture that we will have live, and I look forward to taking your questions then. Thank you.